Today we'll be discussing about microscope. So as we all know that most of the microbes are too small to be seen by our unaided eye, and therefore we require a magnifying instrument that is known as microscope. So microscope is one of the very important equipment in biosciences, and the main function of the microscope is to enlarge the image of the small objects so that it can be seen clearly. There are two parameters, that is magnification and resolution. These are the very important in microscopy. Here, magnification is a measure of how much larger a microscope causes an object to appear, and resolution of a microscope or lens is the smallest distance by which 2 points can be separated and still can be distinguished as a separate objects. Hence, the smaller this value, the higher the resolving power of the microscope and the better the clarity of the image. To resolve microbes and microbial structures of different sizes, we require different types of microscopes. Microscopes are classified based on the physical principles they use to generate an image. So first we'll see the light microscope. All modern microscopes are all compound microscope. That is, the magnified image formed by the objective is further enlarged by one or more additional lenses. So there are different types of light microscope, which include the bright field, dark field, phase contrast, and the fluorescent microscope. These are the most commonly used one. Bright field microscope. The bright field microscope is so called because it forms a dark image against a brighter background. Such microscope consists of a sturdy metal body which is composed of a base and an arm to which the remaining parts are attached. A light source, which may be either a mirror or an electric illuminator, is located in the base. Then two focusing knobs, that is the fine and the coarse adjustment knobs, are located on the arm, and this can move either the stage or the nose piece to focus the image. And the stage is positioned about halfway up the arm, and it holds the microscope slide by either simple slide clips or a mechanical stage clip. A mechanical stage allows the operator to move a slide around smoothly during viewing by the use of stage control knobs. Then the substage condenser is mounted within or beneath the stage and focuses a cone of light on the slide. The curved upper part of the arm holds the body assembly to which the nosepiece and one or more eyepieces are attached. The nosepiece, it holds the 3 to 5 objectives with lenses of differing magnifying power. And this objective lens formed an enlarged real image within the microscope and the eyepiece lens and further magnifies this primary image. The total magnification is calculated by multiplying the objective and the eyepiece magnification. Normally, a microscope is equipped with 3 or 4 objectives ranging in magnifying power from 4x to 100x. and the working distance of an objective is the distance between the front surface of the lens and the surface of the cover glass if it is used, or the specimen when it is in sharp focus. Now we'll see the dark field microscope. In the dark field microscope, an opaque disc is placed underneath the condenser lens so that only light that is scattered by the objects on the slide can reach the eye. The objective lens is positioned in the central region where it completely misses the directly transmitted light, and hence the fields surrounding the specimen appear dark. However, the light scattered by the object, it radiates outward in a spherical wave. Then a sector of this spherical wave enters the objective lens and is detected as a halo of light. In this type of microscopy, as the background is dark and the object is illuminated, such type of microscopy is known as the dark field microscopy. So this dark field microscopy obtain better resolution than the bright field viewing. And one of the most interesting application of dark field optics is the study of bacterial motility. Motility is very important in bacterial diseases such as urethritis, in which the organism need to swim up through the urethra. Thus, dark field optics is needed to detect and observe the helical flagella. A disadvantage of dark field microscopy is that any tiny particle, including a tiny spots of dust, can scatter light and interfere with visualization of the specimen. So, unless the medium is extremely clear, it is difficult to distinguish microbes of interest from the particulates. Next, we'll see the phase contrast microscope. Phase contrast is the most frequently used method in biological light microscopy. In an ordinary microscope, the object is viewed due to differences in color intensities of the specimen, and to create this intensity, the specimen is first stained with a suitable dye which will impart a specific color. Then the differential absorption properties of the stained specimen, it modify the intensity or the amplitude of the light waves which is transmitted by different regions of the cell, and ultimately it creates contrast in the image. Thus, staining is very essential to create contrast in the ordinary microscope, while unstained specimen cannot be observed through an ordinary microscope, and during staining, most of the cells are killed. In contrast, unstained living cells can be observed by the use of phase contrast microscope. Phase contrast microscopy, it exploits differences in refractive index between the cytoplasm and the surrounding medium, or between different organelles. The working principle of this microscope is that small phase changes in the light rays that is induced by differences in thickness and the refractive index of the different part of an object can be transformed into differences in brightness or light intensity. This phase contrast microscope has a condenser which has an annular stop, an opaque disc which is a thin transparent ring which produces a hollow cone of light. As this cone passes through a cell, some light rays are bent due to variation in density and the refractive index within the specimen, and these are retarded by about 1/4 of its wavelength. The deviated light is then focused to form an image of the object. Further, undeviated light rays strike a phase ring in the faceplate, which is a special optical disc located in the objective, while the deviated rays miss the ring and pass through the rest of the plate. If this phase ring is constructed in such a way that the undeviated light passing through it is advanced by 1/4 of its wavelength, then the deviated and the undeviated waves will be about half wavelength out of the phase and will cancel each other when they come together to form an image. The background is formed by the undeviated light is bright, while the unstained object appears dark and well defined, and such type of microscopy is termed as the dark phase contrast microscopy. Phase contrast microscopy is especially important in observing live microbes with transparent cytoplasm, while it is less effective for observing smaller organisms whose cytoplasm has low refractive index. Now we'll come to fluorescent microscope. In fluorescent microscopy, the incident light is absorbed by the specimen and the molecules become excited, which it later release much of their trapped energy as light. The light that is emitted by the excited molecules will have a longer wavelength, thus lower energy. The excited molecules emit the fluorescent light as it gives up its trapped energy and returns to a more stable state. And to observe under this type of microscope, the specimen is usually stained with a dye molecule called the fluorochromes that fluoresce brightly upon exposure to a light of a specific wavelength. In fluorescent microscope, an intense beam of light is produced from the mercury vapor arc lamp or other source, and the heat transfer is limited by the use of an infrared filter. Further, only the desired wavelength of light is allowed to transmit through the exciter filter. A dark field condenser, it provides a black background against which the fluorescent objects glow. The microscope forms an image of the fluorochrome leveled microorganism from the light emitted when they fluoresce. The remaining ultraviolet light is removed by the barrier filter, which is positioned after the objective lens and which could damage the viewer's eyes, or blue or violet light, which would reduce the image contrast. Fluorescent microscopy is used to assess microbial population in a highly dilute natural environments, and they are also used to develop advanced optical systems that reconstruct three-dimensional models of cells. Now finally, we'll come to the electron microscope. So modern electron microscopes, which are in use today, was developed in the 1950s, and they are considered to be an important tool to observe the shape of the macromolecular structures that build up the cells. And the working principle of an electron microscope is that when electrons are ejected from a metal that is subjected to a voltage potential, they travel in a straight line like photons. Like photons, these electrons interact with matter and carry information about their interaction, and they can exhibit the properties of waves. These electrons are focused by means of a magnetic field which is directed along the line of travel of the beam. and as a beam of electron enters the field, it spirals around the magnetic field lines. As magnetic lenses generate aberrations, a series of corrective magnetic lenses is required to obtain a resolution of about 0.2 nanometer. So there are two major types of electron microscope, that is transmission electron microscope and the scanning electron microscope. First we'll see the transmission electron microscope. In transmission electron microscope, a heated tungsten filament in the electron gun, it generates a beam of electrons that is then focused on the specimen by the condenser. As these electrons cannot pass through a glass lens, electromagnets are used to focus the beam. Then the column containing the lenses and the specimen, it must be under high vacuum to obtain a clear image, as electrons are deflected by collisions with the air molecules. Then the specimen scatters electrons passing through it, and the beam is focused by magnetic lenses to form an enlarged visible image of the specimen on a fluorescent screen. Next we'll see the scanning electron microscope. So this scanning electron microscope is used to examine the surfaces of microorganisms. They differ from other electron microscope in producing an image from electrons emitted by an object's surface rather than from the transmitted electrons. The scanning electron microscope, it scans a narrow tapered electron beam back and forth over the specimen. When this beam it strikes a particular area, surface atoms discharge a tiny showers of electrons called the secondary electrons, and these are trapped by a special detector. Further, secondary electrons entering the detector, it strikes a scintillator, causing it to emit light flashes that a photomultiplier it converts to an electrical current and amplifies it. Further, the signal is then sent to a cathode ray tube and it produces an image which can be viewed and photographed. So that was all about microscope, and in the end I'll sum up by stating that the tools of microscopy and molecular visualization, it have shaped the current understanding of the microbial cells, of how they grow and divide, organize their DNA and cytoplasm and interact with other cells.